So here we are, chapter number 2, talking about water. Now, this might seem like a trivial chapter and one you can kind of breeze through. And for those of you who have great memories and can recall everything from largely your Chem 2 class and a little bit of stuff from Chem 1, then by all means go for it. I really wouldn't advise that, but to each their own and everything like that. Okay, so chapter number 2 is water. Okay, now water is the medium for life. Life evolved in water due to the protection it provides from UV light. Organisms typically contain 70 to 90% water. Chemical reactions are going to take place in that viscous solution in an aqueous environment, and water is going to provide that. Water is a critical determinant of the structure and function of proteins, nucleic acids, and membranes. And that's really kind of the key part of what we're going to be getting into with respect to water. And that's really the part of this that's kind of unique. The rest of these things were all almost like barroom trivia or something like that, the 70 to 90% thing. But determining the role that water plays in determining structure of proteins, nucleic acids, and membranes, that's the key part that we're going to get after, or we're going to attempt to introduce with this chapter. Okay, structure of a water molecule. Remember from your Lewis dot structure demonstrations or your Lewis dot structure drawings that the octet rule dictates that there are 4 electron pairs around an oxygen atom in water, and that looks a little bit like this. Okay, these electrons are in 4 sp3 hybrid orbitals, so that means it's 1 part s and 3 parts p. 2 of these pairs covalently link our hydrogen atoms, so we've got covalent linkages here and here. Link our 2 hydrogen atoms to our central oxygen atoms, and the 2 remaining electrons, so there's 4 electrons accounted for, and the other 4 are right here and right here, so that gets us to our 8. Those other 4 electrons are lone pairs of electrons, or non-bonding electrons. Water geometry is the distorted tetrahedron, also known as a bent molecule. Because remember, those 2 lone pairs of electrons, they occupy a ton of space compared to those covalent bonds between the oxygen and the hydrogen. The electronegativity of the oxygen atom induces a net dipole moment. So you have a partial negative up here and a partial positive down here. And that is all going to be, and I'm drawing over everything here. We're going to see better displays of it in just a moment. We've got it basically looking like that. Water can serve as a hydrogen bond donor or acceptor because of this dipole. Okay, so let's go ahead and proceed. There's our water molecule like I drew just a moment ago, and I mean, it looks basically the same. My depiction was, I was limited by the space that I had available. So I don't know. I think that Leninger and company should really consider me for drawing in the next edition of the textbook. At any rate, we've got our lone pairs of electrons. This is a little bit weird of an angle to look at our oxygen atom, but think about it, or our water molecule, but think about this oxygen atom as being far away from us. And those 2 oxygens are here. Oxygen's there, or 2 hydrogens are here, oxygen is here. And then those 2 lone pairs of electrons are occupying as much space as they possibly can. Okay, so hydrogen bonds. Hydrogen bonds are strong dipole-dipole or charged dipole interactions that arise between a covalently bound hydrogen and a lone pair of electrons. Well, if you think about a water molecule, we've got, there's 1 water molecule, lone pair of electrons, and what was our criteria? Covalently bond hydrogen and a lone pair of electrons. Well, this water molecule right here has that lone pair of electrons. Lo and behold, this one right here, this other water molecule, well, that's got a lone pair of electrons, but it's also got a hydrogen bond that can pair up with those lone pair of electrons. They typically involve 2 electronegative elements. Generally, when we're talking about hydrogen bonds, we're talking about nitrogen and oxygen as being those electronegative atoms. Hydrogen bonds are the strongest when the bonded molecules allow for linear bonding patterns. Ideally, the three atoms involved are in a line. And well, lo and behold, if you have enough water molecules, they are able to position themselves and scaffold themselves so that you can have those linear points between our individual our individual water molecules. Okay, so here's a demonstration of, well, a strong hydrogen bond compared to a weaker hydrogen bond. Well, this strong hydrogen bond is highlighted by this water molecule, with these atoms being in a straight line compared to these atoms being at a slight curve. Now you might look at this and say, I don't really understand the difference. I know the first time I looked at this, I was like, what am I missing? And well, if we look, we've got the same players involved here. We've got a phosphorus with a double bonded oxygen, and then we've got this ROH. Well, this highlights strong. Then in our weak hydrogen bond, we've got that same phosphorus with double bonded oxygen, and then we've got that ROH. What's the difference? Well, this one is weaker. I can see that because it's at an angle. But what you should think about is, what is this R attached to or a part of? Well, that R, think about them as being held in position slightly differently. So the stronger hydrogen bond, that ROH, is being held up here, and it's not really able to move, which forces that P double bonded oxygen to be in a certain position so that that hydrogen bond can take place, or that hydrogen bond can be established. Compared to that weaker hydrogen bond, well, that ROH might be a little bit lower, in which case it's closer to, and it allows some movement, some different movement in that P double bonded oxygen. So the strength in those bonds is really kind of dictated by, well, what are the different groups involved here? And that's one of the key parts about, well, protein structure and relating bond strength to protein structure. Because it's not simply these three atoms or these four atoms that are at play. Instead, it's four atoms that are part of this protein, and then four other atoms or two other atoms that are part of this small metabolite or something like that. I hope that made a little bit more sense. Now, hydrogen bonding in water. Water can serve as a hydrogen bond donor or acceptor, as we previously stated, as we kind of looked at with the lone pair of electrons or that hydrogen, that covalently bond hydrogen. Up to 4 hydrogen bonds per water molecule can give it its extremely high boiling point, its high melting point, and its unusually large surface tension. So we have this network that every single water molecule is able to pair up and basically establish a total of 4 different bonds. So 4 different hydrogen bonds per water molecule gives this like very complex network. Now, hydrogen bonding in water is cooperative. Hydrogen bonds between neighboring molecules are as weak as 20 kilojoules per mole relative to the hydrogen-oxygen covalent bonds of 420 kilojoules per mole. So when you think about the strength of a hydrogen bond compared to a covalent bond, it's really important to recognize what that magnitude is. And it's also important to think about kind of this in terms of other types of bonding that can take place. So hydrogen bonds are not particularly strong, but if you think about a water molecule, that water molecule can basically come up with 4 hydrogen bonds. So here's another display of, or here's a display of 2 different water molecules. And what we're comparing here is not so much the bond angles or anything like that, but I want you to look at our covalent, or our bond length. Now this takes you back to Gen Chem 1. Bond length versus bond strength. The longer that the bond is, the weaker it is. The shorter that the bond is, the stronger it is. And we see that here with a hydrogen bond between 2 different water molecules. That's 0.177 nanometers compared to a covalent bond between the oxygen and the hydrogen. There's our oxygen. Oxygen, yeah, I'm having trouble drawing right now. It's 0.0965 nanometers. So it's half as long, and if you compare those strengths, it's 20 kilojoules per mole compared to 420 kilojoules per mole. So it's not necessarily a direct relationship or anything like that, but instead, as that bond length gets longer, well, you see a pretty significant drop off in that bond strength. Importance of hydrogen bonding. It's a source of unique properties of water, as we just mentioned, boiling point, surface tension, melting point. Not mentioned in there, but also worth noting is not surface tension, that was mentioned, specific heat. So the specific heat of water is particularly high. It plays a role, or importance of hydrogen bonds. Hydrogen bonds enable or facilitate or impact structure and function of proteins, structure and function of DNA, structure and function of polysaccharides, binding of substrates to enzymes, binding of hormones to receptors, and matching of mRNA and tRNA. So this right here, that's kind of a very important part of translation, the fundamental process of translation. So going from RNA, mRNA to a protein. So all of the hydrogen bonds are tremendously important. Okay, now biological relevance of hydrogen bonds. We've got a couple of different kind of distilled down simple examples. So we have between a hydroxyl group of an alcohol and water. Okay, so here's our alcohol and a water molecule. Between a carbonyl group of a ketone here, all our functional groups are up top, and a water molecule. Then hydrogen bonding between peptide groups and polypeptides. So this right here is the carboxy termini of, one amino acid, and this is the N termini of another amino acid, both of these found within peptide bonds, or within polypeptides. This is going to impact secondary structure of proteins, which we'll get to down the line. Next up is maybe a little bit more tangible for those of you who have taken a biology class recently, and that is between hydrogen bonding that takes place between complementary pairs or complementary bases of DNA. So the base pair T, or the nucleotide T, thymine, and the nucleotide adenine, A. Okay, so what we have here are a total of 2 hydrogen bonds. Okay, so if you wanted to think about in terms of strength of that bond, that's approximately 40 kilojoules per mole that exists between those two. Now, if you compare that to G and C, well, there's going to be 3 hydrogen bonds between G and C. We're going to get to that in the future, but that's basically 60 kilojoules of energy, or 60 kilojoules per mole. All right.